we go over the process of translation. Well, it all starts from replication. So the first thing is DNA is going to make a copy of itself. So DNA into another DNA. So this is called DNA replication. So the copy of DNA that has been made is now converted into mRNA. Okay, messenger mRNA. And this is a process of DNA transcription. This transcription. Well, now the mRNA have to be converted into proteins, and that is what is called translation. So all this put together is what is referred to as the central dogma of biology. Now our interest is in the process of translation when mRNA is converted into proteins. Now we know that just before translation, during the process of transcription, DNA is found in the nucleus. And so for DNA to be converted into mRNA, this happens in the nucleus. Well, after it does happen, after DNA is converted into mRNA in the nucleus, it will have to be transported into the cytoplasm. Now immediately it reaches the cytoplasm. In the cytoplasm you have got small subunits. Well, what are small subunits? So we have got a ribosome. Now a ribosome doesn't exist together when it is not at work. So actually a ribosome is made up of small ribosomal subunits, and also the large ribosomal subunit. So these two together is what forms a ribosome. So when it comes to eukaryotic cells, the small ribosomal subunits are called the 40S, the large ribosomal subunits are called the 60S, making a total of 80S. So this is a combined. If 40S and 60S combine, they form 60S. Of course, this is not a mathematical sum. 40S plus 60S in mathematics is 100S. This is not a mathematical sum. For prokaryotes, the small ribosomal subunits are the 30S, while the large ribosomal subunits are the 50S, and the combined of this forms the 70S. And so this is simply a ribosome. The small ribosomal subunits, immediately we have got mRNA moving out of the nucleus. Now in the reading in the 5' to 3' direction, the small ribosomal subunits are going to locate the 5' end. Okay, so this is what is called initiation of translation. For the process to start, it all starts with the small ribosomal subunits locating the 5' end. So immediately the small ribosomal subunits locate the 5' end, they are going to start moving. Alright, so they'll start moving along the mRNA in the 5' to 3' direction. So as they are moving, because on the mRNA we have got a set of nucleotides, what is called the genetic code. So the mRNA carries the genetic code. Genetic code is simply a nucleotide, a sequence of nucleotides that codes for proteins, which produces proteins. So let's say this base is adenine, this is uracil, this is, let's say, guanine, this is cytosine, this is uracil, this is adenine, this is, let's say, adenine, guanine, cytosine, guanine, cytosine, uracil, uracil. Let's say adenine, adenine, cytosine, guanine, uracil. Let's say this is a sequence of nucleotides on this mRNA. Now, the small ribosomal subunit starts from the 5' end and moves to the 3' end. Well, immediately they find the reading 5' end AUG 3'. So when they just find AUG, this is going to be a symbol for start. So AUG is called a start codon. A codon is simply a group of 3 nucleotides that signifies that an amino acid has to be brought. So when you are reading the genetic code, we read it in 3.3. There is a video on the genetic code which we can visit. Alright, so if we check, this is the genetic code. So if we have got AUG, we can see AUG is here. It is going to symbolize methionine to be brought, and so methionine has to be taken, and so methionine will have to be brought. Well, what brings methionine is supposed to be tRNA. So the 3 types of RNA always help each other during this process. mRNA is providing the genetic code. tRNA has to bring the respective amino acid, and so tRNA is going to come and attach. So we can see there is AUG there. So at this point, this is where tRNA is going to come. Okay, let me make it like that. Okay, we can see how it is, and so tRNA is going to come. It is in the 3' because mRNA is reading the 5' to 3'. So it's the tRNA which is supposed to come should read in the 3' to 5' direction, and then A is supposed to pair up with U, U pairs up with A, G pairs up with what? With C. So this would be the tRNA. This would be the anticodon of tRNA. So the anticodon tRNA looks something like that. Where this longer side is the 3' end, this shorter side is 5' end. So this region down here is what is called the anticodon. So in this case, it's going to read, okay, there will be a U here, there will be A here, there will be a C there. So this will be the anticodon for the codon AUG. Because AUG is moving in the 5' to 3' direction, that is why this is going to read like this. So the 3' is here, and then the first one on the 3' is that one, U, followed by A, and then C, and then the last one will be the 5' end. So this will be the tRNA which is going to come there. Now I'm going to simplify it all the more, and so that it will be easier to represent. So let's just say this is mRNA, this is the 5' end, and then that is the 3' end. So here we've got nucleotides. So all these which I'm putting are nucleotides. Those are bases: adenine, guanine, cytosine, thymine. Of course, we don't have thymine in RNA, so that will be uracil. And so the one I've put in red is going to be the start, that is AUG. So AUG is a start codon. Whenever we read AUG, then that is going to symbolize start. So we have got our small ribosomal subunits which are moving in this direction. So that is a small ribosomal subunit. Immediately it reaches the start codon, that will be a symbol for tRNA to bring the necessary amino acid. And so let's say tRNA brings that amino acid. There is a tRNA there, it is going to come there. So to tRNA is attached the amino acid. So say this is the amino acid, like that. So immediately tRNA, this is during initiation, immediately tRNA attaches to the start codon, then that will be a symbol for the large ribosomal subunit to come and attach to the small ribosomal subunit. And so the large ribosomal subunit comes in and attaches to the small ribosomal subunit. So this is the large one. So a combination, the large ribosomal subunit is what I'm putting in green. The combination of the large ribosomal subunit with the small ribosomal subunit then forms what we call a ribosome. Well, a ribosome together with the ribosomal RNA, that is rRNA. So it is very difficult for a ribosome to synthesize a protein without rRNA. So a small ribosomal subunit, a large ribosomal subunit, together with rRNA, they are going to start now moving in this direction to now start synthesizing a protein. Now what is going to happen is this compound which is here, okay, so when tRNA attaches to the ribosome, that is called amino acyl tRNA, amino acyl tRNA complex. So when the tRNA complex has attached to this point, the initiation point, that is the point where there is AUG, then this thing will start going like this. Okay, it starts to go like this. Now we have got 3 sites before it even starts moving. There are 3 sites which you should be able to recognize here. So the first site, we can see the first site here before where the tRNA is attached is called the E site. That meaning exit site. The second site here is called the peptide site. Peptide, just like peptide bond. So this is where a bond between amino acids is going to be formed, the peptide site. And then we have got the A site, that is called the amino acyl site. Amino acyl is a combination of, okay, it is called the amino acyl because that is where the amino acid which is attached to the tRNA is going to be brought. So this is where the tRNA is going to arrive. So during the process of initiation, what is going to be brought here will be the amino acid methionine. Well, it is just brought directly into the P site. But as the process continues during elongation, the tRNA is always brought in the A site. But the first tRNA comes into the P site. That is during initiation. But during the next stage we are going to talk about elongation, it always comes to attach in this site, the A site. That is where I call it the amino acyl site. So after this has happened, then the process of initiation has happened already. The process already started of translation. So the next stage would be now away from translation, it now becomes elongation. So we remove that, now becomes elongation. Now during elongation is once the ribosome complex, which we can see the small subunit and the large subunits have already combined, have been formed. So whenever the ribosome complex has been formed, the tRNA that carries the methionine is in the P site, as we can see. The ribosome can now begin to slide along the mRNA to be moving in that direction. Well, as it moves, the next amino acid tRNA that carries the next amino acid is going to be brought into the A site. So this is another codon. There will be another group of nucleotides here. Let's say what is reading there in the A site. Let's say it is CAU. So that means this is red, and then we are going to read on the genetic code CAU. Okay, so that is CAU. CAU is here. So the amino acid histone is going to be brought. So that will be brought by the next tRNA. It is tRNA which is going to bring that amino acid. And so that amino acid is going to be brought. So tRNA is going to come. The tRNA carrying that amino acid is going to come into the A site. So this is the second amino acid, histone. Well, when this happens, once the P site and the A site have been filled, as we can see, an enzyme which is called peptidyl transferase so peptidyl transferase is going to form a bond between these two, which we can see here. So it's going to make that bond. Well, that bond is going to be formed in such a way that this one which is in the P site is the one which is going to join on top there. And so we are not going to see anything in the P site. The amino acid in the P site is going to join to the amino acid on the A site. So I'm going to remove that one. So we can see that that was the methionine in the P site. So the methionine now is linked to histone in the A site. So after this happens, then this complex is going to move one codon in front. Alright, so when it does move one codon in front, what is going to happen? Or let me say 3 nucleotides in front, because we have already formed a bond. So the peptide bond has already been formed. Remember, a bond that holds amino acids together, or a bond that is found in proteins, is called a peptide bond. So in the next step, the ribosome complex moves 3 nucleotides in front. And when we're talking about 3 nucleotides, that means 1 codon. So it moves 1 codon in front, or 3 nucleotides in front. So what is going to happen then? Well, if it does move like that, this complex is going to find itself on the next stage. Let's say the next stage is here. Then the large subunit is on top. Alright, so like that. Still, even when it moves 1 nucleotide, we still have at the start, there is always the E site, the exit site, and then this is the P site, and then this is the A site. So what happens is, it is always moving in the 5' to 3' direction. Let's take note of that. So what was in the P site? The tRNA which was in the P site is going to go into the E site. Okay. So the tRNA which was here in the P site is going to find itself in the E site at that point, because it has moved 3 nucleotides away. So the first one is here, the second one is here, the third one is here. Those are the 3 nucleotides. And then the one which was in the A site carrying the growing peptide is going to go now into the P site. And then we have got this one, and then methionine is still on top there. So once this happens, this one which is in the E site, the tRNA which is in the E site, that is exit site, is going to be released. So we are going to again read the strand which is showing on the what? On the A site. So let's say on the A site is showing GUU. So if it is showing GUU, what will be that? Let's see what it brings. Alright, GUU is here. That means the next amino acid that is supposed to be brought is valine. So valine is going to be carried by another tRNA. So we're going to have another tRNA which is going to come into this site, bringing the valine. So there is the valine. Again, an enzyme called peptidyl transferase is going to come and form a bond between these. Now, a bond always happens in the same direction. So this which is down this tRNA is going to be connected to this amino acid. So the amino acid which is down the tRNA is going to be connected, and so what we are going to have is the amino acid is going to come here, the blue one is going to come here, the red one is going to come here. So the one which was first to be put will always be on top, and then the next one to be put will be always down. So that means this one will go because we formed now a bond. Okay, so the bond is there, so this one has already formed a bond. Even this thing has already evacuated, as we've seen. So the tRNA is now out. It has been removed. So when this happens, again it is going to move 3 nucleotides. So let's do it once more. So when it moves again 3 nucleotides, or 1 codon ahead, that would be hit, is going to find itself in this place. So the ribosome complex has again moved. So what was in the P site, again we have the E site there, we have got the P site on the middle, we have got the A site there. So what was in the P site is going to find itself in the E site, in the exit site. What was in the A site finds itself in the peptidyl site. So the growing strand is always finding itself in the peptidyl site. And so what we have is, that is valine, that is the one which was brought according to the example that we gave. And then we have got that one. And then the first one which we brought in, that is our methionine. So again we are going to read the codon that is going to be in the A site. Now let's say in the A site for now it is reading AAC. What amino acid is going to be brought? So AAC, we go on and read it. AAC is here. So the next amino acid that is going to be brought is that one. So we are going to take now a tRNA, another tRNA is going to bring that amino acid. So another tRNA comes there to bring that amino acid. So it comes here. Again, a peptidyl transferase is going to come in and form a bond. So the one which is down here, the amino acid down, is the one which is going to be connected to this amino acid that has been brought in. Alright. And so the yellow one we can see is going to be there, and then the blue one, the blue amino acid is going to be there, and then the red amino acid is going to be there. So this is going to be out here. Okay. Well, the growing strand continues to go in that order, and it continues to go. Because this is in the A site, this is going to be removed. It is in the A site. Alright. So that is the exit site. So we are going to see what was in the A site. It is now there. That is the tRNA. So in the E site, that is where it is removed. So the strand will continue going and going and going. It will continue being longer and longer and longer. So after that, the process will continue going. Well, we don't need to continue going forever and forever. So that ends the process of elongation. Then you have to step into termination. The process has to be ended. So the last stage is termination. Well, just like we had a codon that specified for the start, AUG specified that the process has to start, we also have a codon that stops the process, and those are called stop codons. The start codon is only one, that is AUG, but when it comes to the stop codons, there are the 3 of them. Alright, when we see, it is even there on the genetic code, whenever we see UGA or UAA or UAG, that is the stop codon, then the process has to stop. So what is going to happen is, this is going to move. It will continue moving and moving and moving like that. So let me just show where it has grown so much. Let's say it is going to reach at a certain point where we are going to have, of course, we have got the P site, the E site, the P site there, and then the A site. So the growing strand is going to rest now in the P site. Now we have got a lot of them which are attached. So let's say we have got this one, there is that one also attached. Next, another one is attached there. Another one is attached. Those are all amino acids. Another amino acid was attached at that point. Another amino acid was attached. Another amino acid was attached. Another amino acid attached. Another one attached. A longer one. Well, the one we know that the one which is supposed to be outside, supposed to be the one which we started with, that is methionine. Okay. So another one attaches there. Then we have got another one like that, and then the methionine which is there. So this strand is continually going and going. So when we reach at the point where we formed a so much long, and then we find a reading here in the A site. So in the A site, when we find the reading either A, okay, so the stop codon which stops this process, as we've seen from the genetic code, either UAA or UGA or UAG, any of these, when they are read, no tRNA recognizes any of these codons. And so what is going to happen is a special protein which is called a release factor is going to come and bind to this site, the A site, because what we are reading is any of these, and we don't have any tRNA which can be able to bring any amino acid. There's actually no amino acid that codes for this, the stop codon, and so a release factor is going to attach to bind there. When the release factor binds to this would ultimately, so whenever the release factor binds to the A site, this will cause the polypeptide chain to break off the tRNA which is in the P site. So that is going to cause this to break off. Okay, so this is going to break off. And so when it does break off, the ribosome complex will again disassociate, and so this will break everything. So we are going to have the small ribosomal subunits alone. The large ribosomal subunits are also going to be alone. They all disassociate, and then we are going to have the growing strand which is going to be also alone. So they are all now broken. Okay, so when this happens, then the process of translation has ended, and so we have got now our polypeptide. So what has been formed here is a polypeptide. Well, at this time, this polypeptide is not functional. So for us to make sure that it is functional, it undergoes some modifications. Or it is a polypeptide, we are not even calling it a protein, because there are some things that have to be corrected before then. Well, let's look at those modifications which it has to undergo for it to be a functional protein. So those are called post-translational modifications. Post meaning it is after the process of translation. Just like when we are discussing the process of transcription, we had modifications at the end, and so even the process of translation, there are modifications at the end. Alright, so let's go on. Now, The ribosome uses the genetic code to translate the mRNA into a polypeptide, as we saw. Once a polypeptide is synthesized, it is usually modified before it actually becomes a mature and active protein. Such modifications are known as translational modifications, which we are talking about. Well, the first modification that what has been formed has to undergo is phosphorylation. Now, what is phosphorylation? Well, certain amino acids such as serine, threonine, and tyrosine found on the polypeptide chain are modified. So certain amino acids, because we have got a chain that has been formed of amino acids. There's an amino acid, and amino acids, all these are attached. Amino acid there. Continually these are attached. So certain amino acids, I'm using AA to mean amino acids. An example that we can give is serine and also tyrosine. So these have to undergo some modification by phosphorylation, and what happens is they are modified by an enzyme which is called kinase. So kinases, these are enzymes which play an important role in the addition of a phosphate. That is phosphorylation. And so this is also very important, because some amino acids are only functional when they contain a phosphate like that. And so we have to add it there to make them functional. So this is one way to make it functional. So that is one post-translational modification. So the enzymes which add phosphates are called kinases. Alright, so that is the first modification that it has to undergo. Now the next modification that these have to undergo is, well, maybe phosphorylation. The process of phosphorylation, which happens by enzymes called kinases, is important. This type of modification plays a crucial role in the cell cycle and also signal transduction. That is the importance of phosphorylation, that is in the cell cycle, and also in the signal transduction. That is the main reason as to why we have to do this. That's why we need to add a phosphate. So for this reason, well, when we are talking about the cell cycle, this is going to be discussed later in the course. Alright, so we are going to discuss that one later in the course. What we need to know about the cell cycle is when we are talking about things like mitosis, meiosis, that is the cell cycle, which we are going to discuss later in the course. Now, the next thing that happens is methylation. Now, methyl groups have to be added also to amino acids by an enzyme which is called methyltransferase. Phosphorylation is the addition of a phosphate, but here we are adding methyl groups. Phosphorylation, we said it is the kinases which do add the phosphate, but when we are talking about methylation, the enzymes which add methyl groups are called methyltransferases. So methylation usually increases the hydrophobic character of amino acids. When we want amino acids to be more hydrophobic, that means there are some amino acids which are not water loving. So such amino acids which are not water loving, to increase their hydrophobic character, we add a methyl group. Why is it that methylation increases hydrophobic character? Well, a methyl is a non-polar compound. Whenever we see a bond between carbon and hydrogen, this is non-polar. So because this is a non-polar molecule, then It is water hating, and that is why it increases hydrophobic nature. All right. Well, this is what we need to get to understand about methylation, and we can as well do more research and dig deeper into details. Now, another kind of modification that happens is lipidation. Well, when it comes to lipidation is the process by which liquid components are added into polypeptides. So, we are saying liquid components added into polypeptides. Now, usually those proteins that are designed to be in membranes, so those which are found in the cell membranes, you want to have lipids on them, alright, such as in the endoplasmic reticulum membrane, mitochondrial membrane, or the plasma membrane itself. So those have to have these lipids, and so that is why they undergo lipidation. So we are adding the lipid components to them, and that is very important. We are saying this is for those amino acids which are used, those proteins which are used in the membranes. And so the reason as to why we are even doing this, adding lipids, well, we are saying lipid components are adding. So the reason as to why we are even adding lipid components is because we want to increase the protein's hydrophobic character. So we want to increase the protein's hydrophobic character. Remember, a lipid has got a lot of carbon to hydrogen bonds, and this makes it non-polar. So the reason of us adding a lipid to this protein which has been formed is to increase its hydrophobic character. Alright. Just as we know, the cell membrane is a bilayer, a lipid bilayer. It has got a portion which loves water and a portion which doesn't love water. And so that is why we want to increase even the protein, because proteins are also used in the cell membrane, and those proteins which are especially used in the cell membrane are what we put lipids on. Alright, so apart from this, another kind of modification that we want to make sure that it happens is glycosylation. Now, glycosylation, this is one of the major ways in which polypeptides are modified. This process involves adding sugar components to the protein. So we have to add sugar components, as we can hear from from glucose. Alright, so this is what happens. Now, why are we even adding these sugar components? So this can affect, just the addition of these sugar components to the protein affects the protein conformation and folding, because the way it is the function of a protein depends on its folding. So we have got different levels of protein structure. The primary structure, which will just tell you the level of, the order of nucleotides. The order of, not nucleotides, but the order of amino acids. We have got also the secondary level, where now the protein starts to fold, where it can fold either into the alpha helix or also into the beta pleated shape. It goes on and goes into the tertiary and quaternary. So folding is very much important, because whenever a protein loses its structure, it has also lost its function. So that is why we are adding sugar components, because they help in the folding process of a protein. So one example of a protein that is glycosylated is membrane proteins, which are receptors. All right, so those are examples, those membrane proteins. These proteins undergo glycosylation. They are glycosylated. So let's look at another modification. Another modification is N-acetylation. Well, what about N-acetylation? So N-acetylation is the transfer of an acetyl group onto the nitrogen on amino acids. So we have got amino acids, and we know that amino acids have got a nitrogen. Actually, an amino acid is in the form C. We have got hydrogen, we have got the R group, we have got COOH, and then we have got NH2, like that. So this is an amino acid. This is the basic structure of an amino acid. And so when we add an acetyl group onto the nitrogen on an amino acid, so I've got a nitrogen there. When we add an acetyl group, what we are doing is performing N-acetylation. Alright, that is why it is even called N, because it is on a nitrogen. So this process can take place when the ribosome is still translating the polypeptide. Just that the discussion we made, just during the process of translation, this is when N-acetylation can happen. Just as during the process of translation, we can add our acetyl group, and at the same hand, it can also happen after the process. It is not just during that. So in most eukaryotic cells, when translation is still taking place, the first amino acid, which is methionine, is usually removed and replaced by an acetyl group. So the first one which we put, as we saw, the first amino acid which was brought was methionine, and methionine is usually removed and replaced with an acetyl group in most eukaryotic cells. So N-acetylation plays a crucial role in gene expression. Well, gene expression is a very interesting subject to discuss about, and we are going to talk about it. So histones, the process that assist in condensing DNA into chromatids, can be acetylated, which reduces the ability to fold and open up the DNA for transcription. So that is why we are even performing this process of N-acetylation. We have said N-acetylation is important in gene expression. Now we are going to discuss about gene expression, because it is also a subject on its own, and we need to understand it much more in detail. So it all starts from gene regulation, where you have got a wide range of things that have to be done by a cell to increase or decrease the production of a specific gene. So when we are forming amino acids, when we are forming a protein, of course, during the process of translation, we need to regulate, we need to make sure that only the right amino acids are put, and so that we need to cut the process. And so to cut the process, this is what is going to happen. That is what is called gene expression in what is called gene regulation. So gene expression, what genes are expressed in the process? What genes are expressed? Are you talking about genes of the air, genes of the what? What are expressed? What are you going to say after the process happens? So these are some of the modifications that a protein has to undergo, a polypeptide has to undergo for it to be a functional protein.